So good morning or good afternoon, depends where you are in the world.
Yeah,
Here in
good
Italy,
afternoon.
good morning. So I'm going to talk about pharmaceutical polymorphs, advanced strategies for solid state characterization and optimization. What are the polymorphs? What is the polymorph research? The polymorph research? Well, actually, it's very important because the polymorphism in pharmaceutical field is so important because of solubility and viability of the solid forms of the drug when the drug is in solid forms. And so we have to study what the physical stability, because different polymorphs has got different therapeutic effects. We will see in a few minutes. So um, one a substance has got polymorphism. One is a solid. That's it. If it's liquid, it has not. And so we study the solid states, the chemistry of the solid state. And when it dissolves, it's not more polymorphism. And what we study? We study not only polymorphs, we study also solvate, co-crystal crystals, and so on, and amorphous also. In this talk, I will talk about polymorphism in particular. Why is so important? It's so important because the development of new solid forms, polymorphs, will give a lot of money to the pharmaceutical industry firms. We will see. One molecular, for example, one substance, I'm talking not about the mixture. I'm talking when you synthesize, for example, you do organic synthesis, you do at the end the final product, and the final product you crystallize, and then you do a different technique to characterize. But from that end, now it is a new beginning. When the drugs is the PA, well, actually not the drug, is a principal is active component, a pharmaceutical active component, compound. Sorry. When this solid compound you have synthesized, now is starting a really new or huge and and a really huge topic, and it's called crystal engineering. So we take the substance, I crystallize in with different technique. Is a different way of crystallization. If you compare to crystallization with synthesis, in organic chemistry, and so after crystallization, it's possible that the solid form will have not just one polymorph, but more than one. And how can we predict? Can we predict that the final forms will be polymorphs? Well, actually not. There are present software for prediction of new crystals, and also there are new instrumentation that you do really a lot of crystallization in few minutes. Well, actually, I do by myself inside the lab. But the important thing is that the best results, when they say is serendipity, it means that you have statistics perhaps when you don't expect, and so from that point we crystallize in different way. I crystallize in different way, and also pharmaceutical firms, they do in really different way. um and at the end of the crystallization, we have to characterize. What does it mean, characterize? Physical and chemical characterization. And to understand what? To understand the physics, the chemical, and the hygroscopy, and the thermal and electrical conductivity, and the chemical stability of the solid form. I thought why is so important. We will see really few seconds. Um, Okay, but you've told me in this talk, I'm talking about the end. We characterize the solid forms, and it's finished. The work is finished. No, the work is not finished. Inside the pharmaceutical firms, they have to do a mapping. It means from the beginning, and this is the beginning in a pharmaceutical firm, and a monitoring control until the end. The end is when the drug, the PA is inside the formulation. So it's called mapping, control from the beginning to the end. When it goes, the formulation, the pharmaceutical formulation goes to be sold. And okay, but it's not important. Someone can think, no, it's really important to do this kind of crystallization and calcination. Why? Because the 60% of barbiturates, the 60 of sulfonamides, and for more or less the 23, 25 of steroids, they present really a different polymorphic forms and solvates. So really important to do this kind of study and research. And why it's so important for FDA? Why? Because it's money. Yes, because before, and they have really rule restriction before undergoing new drugs, new drug in a clinical trials, and before going to the market. So they need monitored production. And also the European Patent Office imposed to anyone it means pharmaceutical firms, to have a patent of a new polymorphic forms. So and patent, it means that you have to crystallize, to characterize with this technique. I'm going to talk about results with one technique. Usually I use more than one techniques. And so what we have to focus, when a substance in a solid forms, it presents more than one polymorphic forms. We crystallize and we characterize, and we have so a complete knowledge of characteristical chemical and physical properties of the active ingredient. And also a new polymorphic forms is not only a patent for the firms, but it can be a patent for another firms. Why? Because remember, solid forms can present more than one. It means that it has got 3, but the firms has studied just 2. Another pharmaceutical firm can crystallize the third forms, and then it can put a patent on the third. And does it can go to the market? Yes, because different. Does it have different therapeutic effect? Yes, it can. So it's so important to study, to have the screening of polymorphic forms, and cause different polymorphs has got different therapeutic equivalence. It means that, what does it mean, therapeutic equivalent? It means that a drug is not a drug. For example, if one polymorph is active and the other no, it means that inside the formulation in the past there will be really a lot of problems about this. A mixture of the two polymorphs, it means that the final formulation didn't cure the persons, and so they have withdrawn to the market. And so how we do the polymorphic screening. Well, actually we do with different technique, not only one. Now I'm discuss about the results with one. We use DSC, different calorimetric scan. We use TGA. We use different techniques, spectroscopic one, not the invasive Raman and IR. and we use X-ray diffraction on powder and also on crystals. If you use crystals, you have to do decrystallization of single crystals, and this is quite difficult to do. And also, I want to use also solid state NMR. But it's important also to study not only at room temperature, to study with different inside also the formulation. I will show you one example about this. So the first results that we had in the past, we study an active pharmaceutical. We study active pharmaceutical ingredients, as I told you, polymorphs, solvated rates, salt, co-crystal, amorphous solid. For example, in amorphous, you can't use X-ray. And then we study in details three different active ingredients. One was pharmaceutical palmitate, the other was tamoxifen citrate, and the other acetylazamide. We will see some of the results with just one technique. This is the formula structure of the first one. Look here, the stable form is for the metastable form is for me, and is a active modification. The first one is inactive. It means that you have no therapeutic effect when it is in your body, and the other one, it is unstable form C. Oh, what does it mean stable, metastable, and unstable? Stable, it means that also inside the formulation, the solid formulation, it doesn't transform itself, and also during doesn't transform itself cause absorbing of water or something else. And with metastable, it means that it can convert from one stable to another, and unstable, really unstable. Here are the first results. So the form A is inactive and then active, and see what we have categorized with the Raman spectroscopy. And Raman spectroscopy, it is like IR, but the bands are from down to up, and we have categorized the bands. It means that these bands we have categorized because it's connected with the ligands like IR. And so we have categorized the meaning of this number. Also, it's possible to heat the solid forms, and it's possible to heat the wall, for example, with this kind of part of instrument added to the Raman instrumentation. And it's possible to heat and also to understand if the heating is transforming for example, in this case, chlorophyll palmitate to one form to another, and it did. It means that at 30 degrees it was form C, at 65 it was form B, and then it was form A cooled. So just to keep in your mind that polymorphs and the heating can transform itself. And we have categorized with this, and also we have done this tamoxifen citrate, and this is form 1 and form 2 were well known. Instead, form 3, but there were not evidence. It means that can we find a new polymorphs? Yes, we did. And then we have categorized with Raman spectroscopy. It means that you have this kind of Raman spectra, and then we have to to study all the band, each band, and study all the band. It means study all the band, also the Raman bands with the really low enhancement of the Raman bands. What we have studied, we're studying inside a melting point fusion, capillary melting point fusion, and it's possible, yes, to study inside the water. It's possible to study decomposition inside inside solvents, and we study through through the capillary. And at the beginning, at time 0, it was form 1, and then after 44 hours, it was form 2. And then also the image, you can see also the image, not only the spectra. And then at 40 and 48, and then at 50, it was just form 2. Just to keep in mind, yes, it's possible to study the crystallization of your substance, of your drugs, and inside with a particle objective, or out in the through the older, and the older it can be made of glasses. Yes, it's possible, and it's possible to have the image and also the spectra. And quite in the end, acetoazolamide, we study form A and form B. We study in this case with FT, ATR, and FTIR, because you have remember you that we use different technique. We have study all the band, and then we study also with Sam to understand what was the morphology after before the grounding, after 5 minutes, and after 20 minutes of grounding of particles. So you can see the image is changing, and it's possible to characterize during the grinding of your drugs with the FTIR, ATIR, or also Raman, in this case FT Raman, how it's changing the enhancement of the bands, and also sometimes the shifts of the band also. So in conclusion, what we can say that it's possible to characterize drugs with not destructive technique like RAM and IR, and also other different technique. We have seen the importance, why it's so important, not only because of different therapeutic effect, and it's possible to crystallize new polymorphic form inside the labs. Perhaps in the next future we'll be very rich and to do a patent of a new drugs. Thank you.